we're going to talk about soil biology today, and I'm going to condense it into beneficial biology. So I'm not going to go down the track and get too deep into mycorrhiza or nitrogen fixing bacteria. We're going to talk about them generally, and we're going to be talking about beneficial biology. So our outline of the presentation, I'll talk a little bit about Tree Preservation Australia, a brief explanation of basic soil and plant health associations, benefits of optimum soil and plant health, importance of root biomass, the microbial connection, and acknowledging, managing key microbiology in a successful soil and plant health care system. All right, so TPA is a partnership between myself and Gerhard Grasser, and we're an Australian entity focused on the practical application of complex soil and plant interactions using leading research and science. So we look at things holistically, the holistic plant healthcare management, consultation, training, specialising in biologically driven maintenance and recovery of stressed and dying trees and their surrounds. But brief history on me. I've been an arborist for about 20 years, and 10 years into it, I went to America, and I was a young fella keen to keep climbing trees and getting all involved in that sort of stuff. And then I was exposed to plant healthcare and looking at soil health holistically. I came back to Australia, started a business in Queensland, and then sort of moved off into consultancy with agriculture, looking at soil health, and then started a business with Gerhard a few years ago. Gerhard Grasser has been around soil health for a long time. He's got another business called Agri Solutions, which is all about empowering farmers and helping educate them to make better decisions and look after their soil health to help their business, their agricultural businesses. So G's been around for quite some time. Now the TPA Methods outline, we're systems approach, it's not singular. We don't believe in silver bullets. There's no such thing as a silver bullet. There's no such thing as a one fix wonder. There never will be in the natural world. The natural world is too complex. So the way we do things is we assess the plant health conditions using our technology data capture. So we take a whole lot of plant health information and soil health information, and that gives us a bit of an idea of what's going on with the plant at that particular time of year in those particular conditions. Then we determine the baselines. So we look at it as it's the new NPK. We talked a little bit more about nutrients. NPK is nitrogen, potassium and phosphorus. But we're kind of learning a lot more these days about how soils actually really work. And we call this the new NPK, which is nutrients, physical structure, and then key biology, the soil biological network. Then we correct those levels wherever we've got problems with the nutrients, the physical structure, and key biology, and we always do. Every time we test soil and we look at trees, we're always seeing issues. Unfortunately, there are a lot of issues out there. Soil health is in really bad shape throughout Australia. And then fourth, we monitor. So we proactively respond to the changes in the soil, the tree and the environment to build the soil and plant health and grow the system resilience. Without monitoring, things can change quite easily. You can have extended droughts, you can have temperature extremes, you can have flooding events, and that can change how the system works. Okay, so into the meat of it. Microbial interactions with trees. So the most important thing to understand is that trees produce exudates in the forms of sugars, proteins and carbohydrates. They release these sugars into the root zone to communicate with the microbiology in exchange for root protection, physical, enzymatic, nutrient, soil building and root navigation. Things like mycorrhiza, that we talked about before, can actually communicate back to the root and tell the root where to go, because the mycorrhiza is out in front. It's sorting out what's going out through the soil. If anyone grows compost at home or makes compost, a lot of people think that worms eat organic material. They don't. It's the bacteria, protozoa, nematodes and fungi that are breaking down that, and worms are feeding on them. So we have here at the top, we've got the trees photosynthesising. It's producing sugars, sugars, proteins and carbohydrates. It's releasing that down into the soil profile to communicate with all of those organisms. So if we start from the top, we're looking at fungi. Fungi primarily are feeding on carbon. Everything on Earth has a carbon to nitrogen ratio. Then we move down into bacteria, protozoa, and then nematodes. And we also have in the middle there root-feeding nematodes. For today's purposes, we're mainly going to focus on all the beneficial groups. I'm not going to go off tangent and talk about pathogens, and at the end we'll talk a little bit about some of the data that we've collected and some interesting things we've found there. So the first microbial group that we're looking at is fungi. Fungi is one of the most important microbial groups. They're all the beneficial microbes are all really important, but fungi are basically the tree's best mate. Without fungi, trees are really struggling. You can see in that picture there a hyphal strand growing. There's a couple of brown bits of organic matter that the mycelium is growing into and feeding off and breaking down, and then you can see soil particles all being held together. Fungi's roles, they're a major plant exudate consumer. So the two main microbial groups that are communicating with trees are your fungi and bacteria. So fungi here, we've got the symbiotic communicator, carbon fixer. They can decompose organic materials, carbon, suppress and fight disease. They can dissolve some parent material, bind soil particles together, improve soil structure, water holding and air capacity, increase root absorption, nutrients and water, and decompose soil toxins. Really important. Bacteria. Here we've got a picture. This is 400. This is under our microscope. This is 400 times magnification. Bacteria, basically, what they lack in size, they make up for in numbers. So bacteria can multiply. One bacterium can multiply to 1 billion in 24 hours. So what they lack in size, they make up for in numbers. Bacteria, same thing again. They're a symbiotic communicator, directly working with the plant and absorbing those exudates, feeding off them and communicating with the tree. They're a nitrogen fixer, like we talked about a little bit before. They can absorb atmospheric nitrogen. So you know when it rains, you can smell the rain event. What you can actually smell is bacteria multiplying. Azotobacter in that case. They're a nitrogen cycler. So bacteria made up of 5 to 1 carbon. So 5 carbons, 1 nitrogen. They are the only organism on Earth that can handle that amount of nitrogen in their body. There's no other organism on Earth that can handle that concentration of nitrogen. They decompose organic material, produce oxygen through oxidisation of water. That's really interesting. I think that paper came out last year. So that's some fascinating stuff. It's not just about it happening in the leaves. They suppress disease, build soil structure, and retain nutrients in the soil, which is very important. Here we have protozoa. This is at 6.4 thousand times. So this is under an electron microscope, some work that G and I did. And this is a ciliate. Now these guys are feeding on bacteria. They're a predator. This guy's amazing. They look like something out of a science fiction movie. You can see there that row of inner teeth, and they basically just cruise along looking for bacteria, find some bacteria, hover over it, and then consume it. Quite amazing watching these guys in action. Actually, and you can just see there the level of what it looks like. You know, with the plates, the titanium sort of steel-looking plates that it's got. They're quite incredible. So we've got 3 groups of protozoa. We've got flagellates, amoebae, and ciliates. They're a bacterial predator. They're made up of 30 to 1 nitrogens, 30 to 1 carbon nitrogen ratio. They cycle nitrogen. They engage in population control of bacteria. If we didn't have protozoa in the equation, bacteria would overpopulate. They mineralise nutrients. They're decomposers. Plant nutrient converters, and they build soil structure. So as protozoa feed through the day, they need to eat 6 bacteriums to make up their 30 carbon to nitrogen ratio. But if they maintained the amount of nitrogen in their body, they'd die. So they release that nitrogen so that they can maintain that 30 to 1 ratio. And then that way nitrogen is cycled. Then our third group, we've got nematodes. This same thing, this is one under our microscope. This is a picture of compost tea. We've got a fungal feeding nematode there with the blown up photo there. We can see the style that they cruise around to try and find fungi, and then they stick their style into the fungi and absorb the nutrients that way. These are a beneficial nematode, and they're quite large. So like we were talking about before with compaction getting high, these guys can't survive in compacted soils. The only nematodes that can survive in compacted soils are root-feeding nematodes, which are not beneficial. Nematodes, again, they're a predator. They predate on bacteria, fungi and other nematodes. Plant nutrient converters, disease consumers. They're an important food source for other larger organisms in the soil food web. They build soil structure, and 80 to 90% of nematodes are beneficial in a healthy system. Okay, so this is an idea of healthy soils. Now, healthy soils it's a bit of an ambiguous term. It depends on where you are, but what's important to gather from this information is that it's about having the groups in ratios and having them there and functioning. So we've got 6 million bacteria and 25,000 species per gram of soil, 150 to 300 micrograms of fungi and 8,000 species, 10,000 protozoa per gram, 20 to 30 individual nematodes, bacterial feeders, fungal feeders and predatory, 200,000 arthropods per square metre, and then 55% mycorrhizal colonisation on the roots. We just don't see numbers like this in all the testing that we do. There's just not a lot of biology happening out there, and it's part of why so many trees are stressed and declining out there. So a healthy soil biological network will suppress disease, retain nutrients. So the microbes will keep the nutrients in their bodies, and as they're there doing their beneficial relationship with the plant root, they'll retain the nutrients in their bodies. And so when you fertilise with inorganic fertilisers and you focus on chemistry, those fertilisers don't stay in the soil profile. They're in a liquid form, so when you get an irrigation event after you fertilise, they wash out of the soil profile. They regulate nutrients. This is important. At the right nutrient, in the right form, at the right time, at the right amount. So when we look at trees or plants, we can't communicate with them. We can only look at visual symptoms that they may have, and we really don't know exactly the amount of chemistry, nutrition that we need to apply. We can have a guess at it, have a crack at it, but I think it's best to leave it up to the microbes and let them sort it out because they're in direct contact with the plant. They decompose toxins for healthier soil function, build soil structure, reduce water use, increase water holding and air capacity, and increase plant rooting depth and root biomass. And they improve soil water access held in the soil cavities, which some roots can't get to. So there's soil water often captured in between soil particles, and the tree roots just can't get to those. But the microbes are small enough to get in between those soil particles and access that soil water in drought conditions or summertime. Healthy soil biological network benefits: water infiltration. Water moves more easily through the soil and through the medium evenly. Air and gases move out of the soil in the right amounts. Nutrient cycling occurs through bacterial and protozoan actions. Disease suppression occurs naturally. Natural protection of the roots and foliage by compatible and beneficial biomass, and increase in plant defence and immune response. That's really interesting with a species called Trichoderma. It's a fungal species. Now, this is a picture of Antarctica. You even have microbes in Antarctica. There's a lot of work going on at the moment with nematologists working down there and understanding how nutrients is occurring with nematodes predating on bacteria. So even in Antarctica, microbes are important. You imagine how important they must be with trees in a forest situation where there's a lot more microbial activity. Now, microbial ratios are really important with trees. Trees are fungal dominant organisms. They're shedding organisms, and they're shedding all that carbon every season for a good reason, to promote fungal dominance in the soil. Grasses, monocots, require bacterial dominance. So if you have a think about in a park where you've got a grass growing up to the tree, that grass is also producing exudates and dumping those exudates into the soil profile but it's doing that to stimulate the type of biology that it wants, not what the tree wants. So when we grow grass underneath a tree, the grass is changing the soil biology, and then we really don't want leaves breaking down over the grass because it spoils the look of the grass, so we rake the leaves up and we take that carbon away every season. We're also taking away the major food source to support fungi. So that's quite an interesting thing. When you're looking at biology with trees, you need to be thinking about, you know, where's my ratio between bacteria and fungi. Bacteria and fungal ratios need to be balanced to meet the specific plant requirements, and replenishment of that organic matter to feed the fungi is crucial to fungal dominant plants. So unless it's going back, you know, every season, you're not maintaining the balance that's required. And then the CDN ratio of the organic amendments is important as well. So when we're mulching trees, you know, all different trees have different carbon to nitrogen ratios. So to stimulate the right biology, you really need to sort of think a little bit more about what type of carbon to nitrogen ratio is in that mulch. This is an idea of the balance that we should have in a healthy system. So this is a healthy forest system, and if you look up there in the top left corner, we've got the root to shoot ratio. So we've got a really good, you know, root to shoot ratio, and this is what we're always trying to go for is more root biomass. We've got a really nice big mycorrhizal fungal block there, and then we've got an even saprophytic fungal block as well. So mycorrhiza is one species of fungi, but it's not the be-all end-all. There's a lot of other fungal species that are also important. We look at the big pool of organic matter there in the brown. That's really important. That's that replenishment of carbon back into the soil. And then we see that the bacteria box is in check. So, you know, our nitrogen cycling, the amount of bacteria that we have and then the amount of predation that we have is balanced for a tree. And then up on the right-hand corner in the blue, you've got all your predators, so all your things that are feeding on everything, and it's a cycle. So that's continually evolving. You know, everything's feeding on each other, and the system is functioning. That's a healthy system. The red box up the top is the plant-feeding nematodes. So they're kept in check. The plant-feeding nematodes are kept in check by the other predatory nematodes. We look at the second box, the second group. This is a poor system. Now this is a system that we see all the time in the urban forest when we do our soil testing. You can see the root to shoot ratio is out of balance. We've got a poor root system. Our organic matter box is smaller, so we're unable to support the saprophytic fungi. This site I can tell is compacted because we've got high ciliate amounts there that indicate compaction, and then we don't have any mycorrhizal fungi at all. So as soon as you compact the soil, mycorrhizae can't survive in there. Our box of plant-feeding nematodes is quite big, and all our predator boxes in the light blue are all small. Now this system is cycling lots of nitrogen, but there's no disease suppression happening, and it's not supporting a tree, and that's why we call it a weed system. This is the sort of system that you see often that's compacted and degraded, and you're really only going to see weed species that are going to flourish in that type of system. So that's what we want to try and achieve in the urban forest. We really need to work on biological health a lot more, because that's what we're dealing with out there. Now, biological succession is really important. Soil microbes, they govern plant succession. So if you look back on the left-hand side of the screen, it's the start of evolution, basically. You can see the blocks of high parent material and that very shallow soil. And then as you move to the right, the soil depth gets deeper and it can support a forest system. So we look at the far end, we've got 700 micrograms of bacteria and 7,000 micrograms of fungi. And you go back to the left and you look, we've got 10 micrograms of bacteria and 0 micrograms of fungi. Now, in the urban forest with the testing that we've done, we're finding that we're pushing biological succession backwards. So our soil health, the practices that we have around trees, is actually pushing succession the wrong way. So for us to maintain a healthy forest, we really need to be aware of that and focus some of our work in that direction to try and maintain a healthy system. This is an interesting photo that I took in Boston. In Boston, they have these suburbs over there where they design them beautifully. They're designed so that you feel like you live in a park. So they build the houses so that you can't see your neighbour's place. They have no fence protocols, and they build in the environment without a really strong environmental impact. But the developers picked up on that because the places are worth a fortune, and they thought that they would just do it their way. So this suburb, they just levelled, and they dozed the whole area. They compacted the soil. They came in, helped them build the houses a lot quicker. They came in, they built all these houses up, and then they brought new soil in, man-made manufactured soil, laid all the soil around, did all the planting, and then after about 20 years, everything started to decline. Most of the plants had filled up the root mass, they'd filled up the soil, they'd exhausted the medium. And there wasn't any biology. We did our soil testing in quite a few areas in this site, and the biological balance was really poor. It had a lot of nitrogen, there was a lot of bacteria in the man-made soil, so there was a lot of nitrogen getting cycled. So probably for the first 5 or 10 years, the trees were probably leaping up out of the soil and growing really well. But as soon as they'd exhausted their soil medium and they didn't have the correct microbiology to help them start to break down parent material to build new soil, the system crashed. So a lot of trees were getting attacked by insects, or they were just declining just because they didn't have enough root-to-shoot ratio anymore. And you can see the compaction lines in the soil profile there. Just not a healthy system at all. Root-to-shoot ratios. Now this is, unfortunately we don't have data that I could find in Australia about root-to-shoot ratios of lots of different trees. Dr. Moore talked about it first of all, but this is interesting from America. The Conservation Research Institute looked at grasses. Now one of the relevant ones for us on the right-hand side is Poa annua. And just have a look in a healthy system, the amount of root biomass that has got. Now that's grass. So you imagine what trees should have in root biomass. If we were able to do a study to really work out all our different species and the root biomass that they have in a healthy system, it would be very interesting. So have a think about the root-to-shoot ratios of grasses, and then think about your trees. Because our trees just don't have the root-to-shoot ratio that we really need to keep them healthy. This is a tree that fell over in the city, and I had a look at it, and it had one structural root there that's broken. It had some signs of disease on the other side, and it just didn't have a very good root system, and I think that's why the tree fell over. So I think when we're looking at trees and we're looking at visual tree assessments, we really need to sort of take into consideration a bit more information about soil health, and start valuing soil health. So back to the whole soil health and biological thing. Trees are producing exudates to feed microbiology. The fungi, bacteria, protozoa and nematodes are all working with the plant, protecting the plant, doing whatever the plant wants, taking molecules, changing them, breaking them apart, making things available that weren't available previously. And when we've got all those microbial groups working and in function, we have worms. So worms are just an indicator that we've got some food source for them there. We've got microbes for them to eat. So that's the... Now this is some of our data that we've got. This is an interesting site, and I'm sorry you might not be able to read it properly, but the reason I put it in colours is because we've got 3 different things going on in this one site. This is a chestnut orchard that sort of wraps around the hill. It's down in Mount Worth, forget the suburb name, and we did our thing there. So we've taken our technology data capture, we've got an idea of where how the trees are all functioning individually, and then we took our pathology data, our biological data, and then our chemistry data. And from those 3 sets of data, or actually 4 sets including the pathology, we can then make good decisions and really get a good idea of what's happening with the soil health. Now, we found Phytophthora. Now that's the red line on the left that follows down, and it follows a natural watercourse, and then it goes off and underneath the road in International Park. Where the line starts, there's a road, and we think that the Phytophthora has come from the road and then moved down. Now the Phytophthora is mainly in that triangle anyway, this is where all the Phytophthora is through there. You can see the trees are a little bit chlorotic. So our first tree, geez, I can't even read it very well. The soil biological activity was really poor. I'll go through the data once we've gone through this. And we had really highly active Phytophthora cinnamomi. The mycologist said that it was one of the most active she's seen for a long time. Now, sample 2 up in the middle, top of the light blue arrow, our soil biological activity was better, was more diverse, and Phytophthora was not active. And then we moved down into the bottom, the dark blue arrow. Our soil biological activity and diversity was the highest. We had really high fungal activity and diversity, and we had 21% endomycorrhizal. So these are chestnuts, so they're endo and ectomycorrhizal. Now, we think maybe because the Phytophthora hasn't spread to that last tree, that the biological activity is actually suppressing and keeping the Phytophthora at bay, because when it rains, that water course works. The water moves. Phytophthora is a water mould. It's not a fungi. It's part of the Amusite class or clan. And actinobacteria are really important with keeping Phytophthora in check and probably consuming it as well. Now we'll move to the data. So we've got 3 sites there. So we've found active Phytophthora, our biological. I don't have time to go through the chemistry or the plant healthcare data, but we'll mainly just look at the biological data. So with our TDC, our technology data capture, getting an idea of where the plant health is at and the soil health showed a major variance in plant health. You can see there from the 2 types of leaf growth. We've got the leaves on the right from the infected trees, and then the trees on the left that weren't infected with Phytophthora. We had some nutrient deficiencies and toxicities from the soil chemistry analysis, but we had major soil biological deficiencies. Now, so this is sample 1, so it was where we've got Phytophthora around that tree. Now you can see there with active bacteria. Can you see it? Not very well. Okay, so we've got up on the top line on the left, we've got active bacteria. We've got 0 active bacteria. Our total bacteria was 5.2, so it's just in range. We've got 0 active fungi as well. Our hyphal diameter is a little low. We've got 7% endomycorrhizal colonisation and 0% ectomycorrhizal colonisation. And we've got really low amounts of protozoan nutrient cyclers. So if we've got low bacteria, they're the main food source for protozoa, so your protozoan levels are going to be low as well. Now, what's really interesting is this is the area where Phytophthora is active, and we have 0 active bacteria and 0 active fungi. So, there's no biologicals there to actually, keep the disease at bay, or consume it as well. Some of the other groups we've got there, we've got bacterial feeders, fungal feeders, but they're all in fairly low amounts. But the main important information to gain out of that is that we've got 0 active bacteria and 0 active fungi, the 2 main microbial groups to help plants survive. Site sample 2, which was up on the top of the ridge where we don't have Phytophthora active, a little bit better. So we've got active bacteria up on the top left. It's just in range. Our total bacteria is just in range. Our active fungi are below range, and our total fungi, our active fungi are below range, and our total fungi are in range. We've got less mycorrhizal colonisation in that area, and all our protozoa groups are all fairly low as well. So that system's functioning a little bit better than the previous one where we didn't have Phytophthora. And there was a little bit more organic matter up the top there, but we've got more on the 3rd slide. The 3rd slide is sample 3, so this is where we've tested. This was on the right-hand side of the arrows, the dark blue arrow. We've got in range, we've got better active bacteria, better total bacteria, and then we've got above range of active and total fungi. So we've got a lot of really important fungal groups that are communicating with the plant and attacking disease. And our hyphal diameter is wider there, which is suggesting that we've got beneficial fungi. That system is working better. We've got 21% endomycorrhizal colonisation, and we've got 0 ecto again there. Now, from here, the trees weren't. I'll go back to the other. So the blue arrow on the right, the Phytophthora is it's not going anywhere from that area. And we're suggesting there that it's the biological increase in biological activity that's maintaining the Phytophthora from spreading further. So I think that's about it. So we'll just reiterate how important soil biology is. The plants are producing exudates. They're dumping those exudates out into the soil profile to communicate with the fungi, bacteria, and then we have protozoa and nematodes that are feeding on the bacteria and the fungi, and the system is working, and we've got a really nice root to shoot ratio there. So yeah, I think that's it. That's it.